Welcome and thank you for joining us for today's webinar. My name is Rob Brazis. I'm the Next Gen Sequencing Product Manager here at Lucigen Corporation. And I wanted to start by letting you know that we are an official distributor of Sygnis products in the United States. And as such, we invited our colleagues from Sygnis to present a webinar on whole genome amplification using their TruePrime WGA kits. And so without further ado, I'd like to introduce Angel and Armin, who will present on that technology in today's webinar.
Methods for whole genome amplification can be grouped into two main categories. In the upper part of the slide, you can find the methods based on PCR, while in the lower part there are the ones based on multiple displacement amplification. Multiple displacement amplification is based on the combination of Phi29 DNA polymerase and synthetic primers. First of all, the primers bind to different sites in the denatured input DNA. Those primers are extended by Phi29 DNA polymerase, and the enzyme is able to displace preceding strands, generating new single-stranded DNA where new primers can bind and be extended. This is how the reaction is exponential but isothermal. The main features of Phi29 DNA polymerase are: it has an extreme processivity, it shows a high speed of synthesis, it shows a potent strand displacement activity, and also a 3' to 5' exonuclease stop reading activity, leading to an extreme fidelity of synthesis. This table summarizes the features and perceptions of both categories, having each category advantages and disadvantages. For example, MDA methods show a very low error rate and a very good coverage, but the allelic dropout rate is high. At the end of this webinar, we will add TruePrime to this table, and you will see how it combines the best from both groups. In TruePrime, random primers are substituted by an enzyme called TthPrimPol that generates the primers during the amplification reaction when they are needed. Firstly, TthPrimPol binds denatured DNA at different sites and synthesizes short DNA primers. Those primers are recognized and extended by Phi29 DNA polymerase. And then Phi29 polymerase is able to perform strand displacement. And in those displaced strands, TthPrimPol can bind again and generate new DNA primers that are again extended by Phi29 polymerase. So basically, the reaction is performed the way that in the cases that use random primers. But the main difference is that the primers are not present since the beginning of the reaction. They are generated during the reaction. And this difference results in many advantages. This project began in 2013 when human PrimPol was discovered and characterized. In this slide, you have a list of properties and roles of this enzyme in the human cells. However, this enzyme has stability problems that impede its use for biotechnological applications. Human Prim1 contains conserved motifs, A, B, and C, that form the enzyme active site. The already characterized PrimPols also contains the 3 conserved motifs, but also a thin finger domain, here in pink, required for primers activity. We found a putative PrimPol in Thermophilus that contained the 3 conserved motifs, but lacked the thin finger domain. However, we performed a detailed enzymatic characterization and found that it has all the properties described in members of the family. The features of TthPrimPol as a primers are that it can use either manganese or magnesium as metal cofactors. It shows convenient wide sequence specificity, but prefers CTC as site. It can use ribose or deoxynucleotides at the 5' position, but only deoxynucleotides at the 3' position. So it is also DNA primase. In this experiment, we combine primers or PrimPol with Phi29 DNA polymerase to amplify 1 nanogram of human genomic DNA. As you can see, PrimPol is able to find linear DNA for its amplification by Phi29 DNA polymerase, making unnecessary the use of random primers. Here you can see the amplification products obtained using as input the DNA amount to one human cell. 6 picograms. You can see the greater producibility among independent samples and the absence of background when input is added to the reaction. In the right part, you can see the yield you can get depending on the incubation time, depicted 3 or 6 hours of incubation. One advantage of TruePrime is its extreme sensitivity. When the input amount is limited, between 1 and 100 femtograms, TruePrime shows better amplification yields than reactions based on primers. Moreover, the amplification products are always target-derived, even using very low DNA amounts. We perform the amplification of 1 nanogram, 1 picogram, or 1 femtogram of human genomic DNA, and sequence amplification products in an ion torrent. As you can see, in all the cases, the majority of sequences match the input in the reaction, even when using only 1 femtogram of DNA as input. Another advantage of TruePrime is the primer artifacts. In this experiment, we amplified 1 picogram of human genomic DNA with TruePrime and with random primers, and sequenced amplification products. As you can see, when using random primers, almost 20% of the sequences are unknown due to the primer-dimer amplification events, while with TruePrime, more than 99 match the input. Now, my colleague Armin Schneider will show you the amazing deep sequencing results we have obtained with TruePrime. Thank you for your attention.
Okay, thanks, Ansel. My name is Armin Schneider, and I'll guide you through a single-cell sequencing experiment we have done to look at a lot of coverage details. I have to say this TruePrime technology is available in 3 kits so far. One is a general whole genome amplification kit, one is a kit for amplification of single cells or a few cells, and one is a kit for amplifying circular molecules called a rolling circle amplification, which is also extremely sensitive. And we'll come back at the end of this presentation to how you can order or access these products at the Lucigen website. So as an introduction, of course, I guess most of you are interested in this webinar because you're interested in single cell sequencing, and this is from a review in Nature where the basic advantages and disadvantages of single cell sequencing are summarized. On top you see the standard way of analyzing genomic DNA in biopsies or tissues or in cell culture, and you simply take a bulk of cells and extract DNA, no problem, and you have tons of DNA and can generate nice libraries and sequence those, but doing so will give you nice sequences, but you will have a huge averaging effect. So if you have mutations in just a few % of the cells, you can likely miss those in this approach. So therefore, people are turning towards single cell sequencing, and this is summarized here at the bottom. You take one cell, isolate one cell by any number of techniques that are available, and then you have to amplify DNA from the cell. And you get, in the end, can generate a library and get DNA information from this one cell. The critical step in this is, of course, the amplification of the DNA, because this will lead to bias in the sequences you will finally obtain. And, of course, none of these methods around is perfect. But we think we made great improvement in getting rid of a lot of bias in this amplification process. So here again you see an example from a review in oncology, where you see that a lot of data from different labs in the last 5 years have led to the realization that in tumors, the heterogeneity between cells and clones in this tumor is far greater than has been previously anticipated, and therefore it's of extreme importance to have the single cell sequencing possibility to fully analyze all the clones in one particular tumor. So to look at the sequence coverage and variant detection in amplified single cell, we took single human hex cells as an example, isolated single cells manually and verified that we had one cell. And then we compared basically non-amplified DNA from the same hex cells, so just from a bulk of cells where we extracted a lot of DNA. This sample is also abbreviated as NA in the following. And then we sequenced 4 cells using TruePrime amplification for 3 and 6 hours. We compared this to 2 random primed NDA protocols, one which we call here generic random primed NDA, and this is basically where we use the same, pretty much the same buffers and the same Phi29 enzyme we use in our TruePrime method, and just replace the PrimPol enzyme with random hexamers. And we also compared this to one commercially available random primed NDA protocol, which we call competitor R here. And on the left-hand side, you see the isothermal reaction process. It's basically quite simple. You add your cell in 2.5 microliters of PBS or water or any kind of buffer you use, and then you lyse and denature those cells for 10 minutes, and then you add all the neutralize this thereafter, and then you add the whole ingredients of the amplification mix and let the reaction sit at 30 degrees Celsius for 3 to 6 hours. Of course, if you can do with less DNA than 2 micrograms or so, then you can also go down with the incubation time. So we performed Illumina sequencing at a service provider using just standard covariance fragmentation and standard library preps for all those samples in parallel, and sequenced those with an Illumina HiSec and with a read length of 125 base pairs and a paired read approach. And we analyzed this by commonly available tools such as the CLC Bio Genomics Workbench and the Illumina Base Space, and some dedicated tools for CNV discovery. So this is basically the 3 comparisons I will show you. On the left-hand side is what we call the competitor experiment. Here we compared exactly the same read number we put into the analysis of the non-amplified sample, the generic NDA protocol, the commercial NDA protocol, and our TruePrime amplified cell. Then we have the reproducibility experiment where we simply compared the 4 samples, 4 cells amplified with TruePrime to the non-amplified DNA, and one DeepSeq experiment where we sequenced 3 of those samples to a coverage of approximately 20x genome coverage. So first we'll go to this competitor experiment. And just as a remark, I'll introduce some terms here most of you will probably be familiar with. When looking at coverage, there's basically 3 parameters to look at. One is the coverage depth, that is how many reads can be aligned over one particular base in the genome. Then there's coverage breadth. That's really important. What fraction of the genome can be covered at what coverage depth. And then there's coverage uniformity, which is also a very desirable property, as we will see. And one way we use to look at coverage breadth is to compare this to the maximally possible coverage by using a Poisson distribution formula that was described by a paper Lander Waterman from 1988, and this nicely simply gives you the maximally possible coverage breadth you can obtain with any number of reads. So we did this. We compared the maximally possible coverage with the reads that actually align to the human genome, and those are given in orange here. So we see for the non-amplified DNA at this input and the number of reads that actually align to the genome, we would expect a maximally possible coverage of 59%, and actually we reached 53%. So that's quite a good value. It's a 10% deviation from the maximally possible coverage, and of course this is due to bias within the library prep and the sequencing itself. With TruePrime, we expect 57%, and we get 49%, and this is a deviation of 14%. So this is still a very, very good value, quite close to the non-amplified DNA. For both random primed NDA protocols, we see a deviation in both cases of 35% from the maximally expected coverage. So this is quite some distance from the TruePrime protocol. And we just graphed this, these assembled genomes into a circus plot. And you see here the blue outer ring here is the non-amplified DNA, and you see this looks very nice, very smooth for all the chromosomes, and you can also see nice the aneuploidic parts in the hexel genome. So this looks very smooth. The next one in green is our TruePrime amplified cell, and we see in general that this looks a little bit more rough here, a little bit more peaky here, but in general it follows nicely the pattern of the blue ring here. The next one is the commercial random prime MDA protocol, and you immediately see that there's much more bias in the coverage pattern. This looks much more uneven, and there's a lot of spikes in here. Also, the same holds true for the generic random primed protocol, where you also see this spikiness. And we can also compare the relative chromosome coverage between the non-amplified sample and TruePrime. In blue here, you see the all the chromosomes from 1 to the X chromosome, with their relative coverage at this 12 million base pair input. And you see there's some difference in between chromosomes and the relative coverage. This owing to the sequencing process itself, or to the aligner that has problems with some parts of different chromosomes. And you see in general that the TruePrime amplified sample follows mostly the patterns seen already in non-amplified DNA, with some examples for some of the chromosomes where there's a relatively lower coverage, like for example here chromosome 19. We looked at GC dependency of read frequency, and You see here the plot. We expect a peak here at about 40% TC content for the human genome, and we see this. In blue is the non-amplified sample, and in orange we see the TruePrime sample. In general, both curves look quite similar. If you use methods that have higher resolution, you see here that there's actually a small bulb here for the TruePrime sample, and this shows a small preference for a GC content between 16 and 24%, but overall the impact of this little bulge here is negligible. So next, we looked at reproducibility in those 4 cells, and we have amplified in the meantime many more cells where we also see this nice coverage pattern. Again, you see the non-amplified sample, the outer ring here, and 4 cells amplified for 3 hours or for 6 hours with the TruePrime method. And you see that all of those generally have a nice coverage pattern, and also all parts of the genome are hit, so there's no major part missing in any of those cells. On the right, you see a view on just chromosome 3 as an example, where you have a higher magnification, and you see how similar these the coverage pattern looks in between the cells and in comparison to the non-amplified DNA. Next, we looked at CNV detection, and we used a tool called Ginkgo that's freely available via a website, and it's based on a paper that appeared in 2015 in Nature Methods from the Schatz group. And this is a very nice tool. It's specifically designed for comparing single cell amplifications. And we compared for samples, non-amplified DNA, TruePrime, the generic protocol, and the commercially available protocol. And you see in the non-amplified DNA, there's very little dispersion of the bins that are used to generate the relative copy number over the whole genome, and you see that there's some quite complex over amplifications of parts of chromosomes that happen in the hex cells. And when we look at TruePrime, you see that in general there's a little bit more dispersion of those of the bins used to generate this plot. And in general, the same pattern is also seen in the TruePrime sample. However, some finer details are not detected, such as this very high over amplification here. When we look at the 2 random prime protocols, we see that there's much higher dispersion of the reads, and a lot of the pattern, especially in the last sample, is lost here. A lot of the copy number variation is lost. So this is actually known, as Angel said at the beginning. This is one known weakness of the current MDA protocols, that offer this. There's very nice coverage breadth of the genome. There's a high unevenness in the coverage pattern observed, and this makes CNV discovery particularly hard in MDA methods so far. And this is also highlighted here on the next slide, also taken from the Darwin paper, where the authors have compared literature data on single cell amplification based on 3 different methods: PCR in blue here, the Malbec method in orange, and in green the MDA methods. And they used a novel measure they introduced for measuring dispersion in between the bins used for generating and deducting copy number variations, and they call it MAD, median absolute deviation of all pairwise differences in read count between neighboring bins. And they come to the conclusion, as is always already known in the literature, that the PCR-based methods have the lowest dispersion and they therefore are used for preferably for CNV detection. Of course, they have other problems, like the coverage breadth is quite poor, and the fidelity is not as good. But for CNV discovery, these methods are quite good. And we also calculated this value for our TruePrime amplified cells here, which you see on the left-hand side, and we come up with the same MAD value, that is 0.2, as the PCR method. And you immediately see that this is a huge advantage over the current MDA methods that have a quite high dispersion, making it quite hard to use MDA for CNV detection. So this is a huge improvement of the TruePrime technology, owing to the much greater evenness in the coverage pattern observed. Another problem that is ascribed to the MDA methods is chimera formation. Of course, it also happens in the PCR methods, but there's some assumption that chimera formation is a particular problem in the MDA techniques, going back to a paper from Lucigen. And one of the main mechanisms that is thought to result in chimera formation is the strand displacement process, where during this displacement of the strand that runs in front of the polymerase, there could be template switching, and this could generate inverted repeats or more complex structures. We have used the paired read sequences and measures to estimate the degree of chimera formation by looking at broken read pairs that are due to a wrong distance or mate inversion, so that events that could happen during the strand displacement process. And we see this is for the non-amplified DNA at about 2.5% of all read pairs. And for all the TruePrime samples, we see that there's actually an increase. This looks, the absolute percentage is between 4 and 6.5%, and this is also at the same value for the competitor R here for the commercially random prime MDA protocol. So we estimate that there's an absolute increase of about 3% of those broken read pairs due to chimera formation, and we think that this does not pose a really relevant problem, at least not for any resequencing approaches. Finally, I think the most exciting data is on the deep sequencing experiment, where we compared non-amplified TruePrime and one commercial protocol. And we looked at the total coverage breadth we reach at a very high coverage depth. So here if you just look at the rows at the bottom here, we reach a what we call here a true coverage of about 19.2 x genome coverage with the non-amplified sample, meaning that these are actually read pairs that are correctly placed onto the genome. So this is real coverage. And for TruePrime, we reach about 19.6, and for the commercial protocol, 20.5x real genome coverage. And with those values, we cover 91.6% of the genome in the case of non-amplified DNA and 91.26% in the case of TruePrime, and 85.6% for the commercial protocol. So we are quite close to the value reached in the non-amplified DNA, and this gives us an even better coverage breadth than the random primed MDA methods so far. Also, of course, this coverage is also not bad. And both as Angel said, we know that the fidelity of this process is very high. We also see this in our sequencing results. There's hardly any increase detectable in the error rate compared to non-amplified samples. And we see that the coverage breadth is really excellent with the TruePrime method. So these are two parameters that would allow us to do a very nice single nucleotide variant discovery. And we did this using these deep sequencing data. And you see here on the left-hand side the non-amplified sample. These are all SNVs detected. We used a very sensitive algorithm from the CLC workbench and compared this to the TruePrime sample. And we looked at the overlap here between the SNV space between both methods, and we detect 3.3 million SNVs in common in both samples. And the parameters, the heterozygote homozygote ratio of 1.27 and the transition transversion ratio of 1.8 point towards that these are most of these are real SNVs. So this is a really good value, I think, for this amplified single cell DNA. We detect about 3.3 million in total, and the space that lies outside this overlap here in both samples, the parameters in this outlier space point towards that these are all false positive due to the very high sensitivity of the method we use. And you see this here, the novelty rate in those SNVs is really high, 87%. So this is all likely false positives. The same with the non-amplified sample. When we compare this SNV space from the non-amplified sample to the commercial MDA protocol, we see that there's the overlap is much smaller. It's only at 2 million SNVs that are common, also with good values. And the outlier space is relatively much bigger. So we have detected with TruePrime, we think a quite complete fraction of the SNVs. Finally, the one very interesting parameter that is always discussed is the so-called allelic dropout ratio, and this means that during the amplification process there is preferential or exclusive amplification of just one allele in a number of cases, and this leads to the generation of apparently homozygous variants from real heterozygous variants. And this conversion ratio of heterozygous SNVs to apparently homozygous SNVs can be used to estimate this allelic dropout ratio. And we have done this for all the SNVs we have detected in the genome. And you see in this plot this apparent conversion rate from heterozygous to homozygous variants, and you see that there's there's some variation across the different chromosomes. But overall, over the whole genome, this conversion rate is 2.5%, and this leads to an estimated allelic dropout rate of about 5%, since we only observe one of those events, one of the homozygous SNVs in most of the cases. So this is a really good value. The commercial random primed NDA protocol has a much higher value of more than 40%, but of course this is also due to the higher unevenness in the amplification pattern observed, that will lead to a high number of SNVs that have a relatively lower coverage and thereby allow false calling of homozygous alleles. So in summary, coming back to the initial comparison that Angel showed to you, if we now place TruePrime besides the MDA-based and PCR-based protocols, we see that the TruePrime preserves the very high fidelity of the protocol, leading to a very low error rate. Of course, the fragments generated are very long. We have seen that the chimera formation is not a problem, but it's also another problem in the random primed NDA. As we have seen, the completeness of genome coverage we think is really excellent with this method. We have a lot less bias, a lot less inequality in the coverage patterns observed. So this is a real advantage over the random primed MDA, and this leads to a very good possibility for CNV detection, which has been a previous weakness of the NDA protocols. And the allelic dropout ratio rate is, we think, really small, with 5%. Literature data for the NDA methods range between 5 and 50%. So far. There's a high variation due to the different methods used and so forth. So this all results in a very good coverage breadth, the very low inequality of amplification, and the high fidelity results in a really and the low allelic dropout rate results in really excellent SNV detection, so we think that TruePrime keeps key advantages of the NDA-based method, but adds a few properties where we have improved the NDA protocol to make it really suitable for variant detection, both SNV and CNV detection. And these two products of the TruePrime line are available via Lucigen in the United States. And one is this single-cell whole genome amplification kit, available in a reaction size of 25 and 100. And one is the general purpose WTA kit at the same reaction sizes. And you can visit the Lucigen website for additional information, or directly write an email to the tech service at Lucigen with this address here at the bottom. Okay, and that's it from us, and we are waiting for any questions from you. And I'll hand over to Rob again.
All right, Rob here, Lucigen. Thank you very much, Armin and Angel. That was great, super informative, and fun to listen to. So does anybody in the audience have any questions? If you do, I appreciate it if you just type them in the chat window, and we'll see if we can answer them for you. While we're waiting for folks to type in some questions, I should let you know that Next month, I believe, we have our next webinar that'll be coming up, so keep your eyes open for it. It's going to be a discussion on the challenges of library preparation, both for fragment DNA libraries and for mate pair libraries. So look for an invitation to come for that soon. So the first question, can I use crude samples as input for TruePrime WGA, or do I have to use purified DNA?
So actually we have limited data on this, I have to say. We assume that for the WGA protocol, the TruePrime process is not very vulnerable to inhibitors or contaminants that come in from the DNA sample, because first of all, this will be rather small volume, and second, the PrimPol enzyme is incredibly stable in its performance, but we have not no real data on this. We have done most of our amplification with purified DNA, but I think it's worth a try if you have just crude samples to try to do this. I think this would work.
Okay, super. I have another question while we're waiting. Is it possible to use purified DNA as input into the single cell kit? If I had the kit around, could I put purified DNA in there instead of cells?
Yes, you could.
Okay.
Yes, you could. I mean, of course, there's specific buffers in there that would break open the cells and denature the DNA, and you don't really need all the components. But you could also just use pure DNA if you have this, and run it through the protocol, and this would likely work as well.
Okay. Anybody else? Another one. What precautions should be taken to avoid contamination when setting up single-cell amplification? Great question.
That's really a very good question, and that's and actually we produce these kits, and we take incredible amount of precautions to produce these kits in a non-contaminated form usually it's best if you use a laminar flow to perform the experiments, and all surfaces have been cleaned before, and you UV irradiate all surfaces, and you use only certified DNA-free pipette tips, tubes, and whatever. And if there are any, if there's any plasticware that you cannot buy certified DNA-free, then you should UV treat these plastic or plasticware before you use it. And for in the laminar flow, of course, you use gloves, and it's best if you have gloves that reach up to the elbow, so there will be little chance of bringing in any stuff that adheres to your lab coat or so. So this will be the optimal procedure to do this.
Okay, super, thank you. Just for anybody asking questions, it looks like some of you are asking questions, but you may not have the all recipients checked on the bottom of the chat window. You need to do that in order for, I think, everybody to see your questions, including me, so I can't repeat them. So if you have any questions out there, please double check your, here we go, yay. All right, now they're coming through. Make sure that the all is selected. So next question, which is the sequencing depth of the experiment with HEK cells? 91.26% of genome coverage? Question mark. Sequencer. Okay. So I guess the question is, what was the depth of the 293 experiments?
So we, I think it was 19.6 X genome coverage.
So depth of coverage was 19x, and the overall coverage was about 92%, correct?
Right. With this real coverage depth, you reach about 91.26% of the whole genome.
Okay. Anybody else? Oops, still somebody typing. We'll give them another minute or so. Okay, here you go. You have similar length of amplicon as commercial MDA. Of course, this is, commercial MDA. What are the main reasons that make TruePrime WJ more uniform in genome coverage?
So we indeed have a quite a similar length. The fragments amplified are a little bit lower in length than the random primed MDA, and the main reason that, pardon me? The reason why the coverage pattern is much more even with the TruePrime method lies in the priming process. We believe that there's bias introduced by the random hexamers, and that the PrimPol leads to a much more distributed generation and priming event during this process.
All right. Well, it looks like we don't have any other questions coming through. So thank you very much, Armin and Angel, for doing the presentation for us today. And if anybody in the audience has questions, additional questions, don't hesitate to contact our Lucigen tech support team. You can email techserve@lucigen.com, or in the US, you can call the toll-free number. So thank you very much again, everybody, and take care and have a good rest of your day.